Hello to our friends joining us via recording. We started our class today with some group work to talk about the different kinds of bonds that we did in class yesterday. So a quick reminder for us that the three kinds of bonds we talked about in class yesterday were called polar covalent bonds, and we had nonpolar covalent bonds, and the last kind we talked about were hydrogen bonds. So our goal with the activity we see here is to figure out which kind or kinds of bonds are described by each of our statements here. So I know when I was talking to one of my groups that this one was a little bit tricky for us. So let's skip this one. Let's work our way down the list and we'll swing back to this top one up here. So the second statement on our list here asked about the kind of bond that generates dipoles. There's one kind of bond that I use to make dipoles. Which of these three kinds of bonds up here was the one that makes dipoles? Yeah, so the one that makes dipoles is the type of bond called a polar covalent bond. A polar covalent bond. Here's a quick terminology review for us. When I use the word dipole, what a dipole is an area of partial positive or negative charge. Partial positive or negative charge. The emphasis here being on the partial. The way that I get a partial positive or negative charge has to do with the way that I share my electrons. If I have a partial charge, partial positive or negative, or in other words, if I have a polar covalent bond, do I share my electrons equally or not equally? If we've got these dipoles, if we've got partial charges, equal or unequal sharing? Yeah, so a couple of us are chiming in. When we have areas that are partially positive or partially negative, that's because we have unequal sharing of our electrons. Remember, electrons have a negative charge. If electrons spend a lot of time around one of my atoms and not as much time around the other, we end up with areas that look kind of negative or areas that look kind of positive. That's what a dipole is. So dipole is somewhere that looks kind of positive or kind of negative. And the reason I get those is because we're not sharing our electrons equally. When we don't share electrons equally, we have what's called a polar covalent bond. Now, my question over here on, let's see. Oh, do I not have that? So when we talk about, so I put unequal sharing here then. What's the name of the type of bond? What if we were talking about equal sharing? Which kind of bond is an equal sharing bond? Yeah, those are the non-polar ones. So we'll make a little space over here because I forgot to ask that. Non-polar covalent bond is an equal sharing bond, an equal sharing bond. So remember from class yesterday, we talked about this word covalent means sharing. Covalent bonds are sharing bonds. We can do it equally, which is a non-polar covalent bond, or we can do it unequally. That's a polar covalent bond. So for my question over here that talks about sharing electrons, both of my types of covalent bonds would be considered bonds that we share electrons in. So the correct answer for this one would be polar covalent bonds and non-polar covalent bonds. Down there. Remember when we were talking about hydrogen bonds, I told you guys to consider putting that word bond in quotes, right? There's my little air quotes. Hydrogen bonds. The reason that we're putting them in quotes is because in hydrogen bonds, we don't actually share electrons. When I talk about forming a hydrogen bond, can anyone describe for me in the chat, how did I form a hydrogen bond? What was happening? with a hydrogen bond. It's not sharing electrons. We're frantically typing. Or does anyone happen to see on our slide here, there's actually a description of how hydrogen bonds form. Anyone want to grab that pencil and circle it? Yeah, so a couple of us are typing in the chat. Yeah, when we're talking about, there it is. Yep, there's our circle. When we're talking about dipoles being attracted to each other, that's the way a hydrogen bond forms. So dipoles being attracted to each other. Dipoles are what we see forming our hydrogen bonds. So hydrogen bonds form when two dipoles are attracted to each other. Now those dipoles are these partial positives or negatives. So when we're talking about a hydrogen bond, hydrogen is always a partial positive. Remember, we said yesterday that what it's usually attracted to is the partial negative in things like oxygen or nitrogen. So hydrogen bonds form because we have those dipoles. Those dipoles formed because we had polar covalent bonds. Let's bounce back over to my statement over here. When we talk about playing nice with water, playing nice with water means, in other words, the way that we talked about it yesterday was this would be a compound that surrounds itself with water. That's what I mean by playing nice with water, because we've got listed over here something that does not play nice with water, so pushes water away. That might be another way to think about playing nice or not playing nice. So when I talk about the kinds of bonds that I'd find in compounds that do play nice with water, what did we think on that one? Who plays nice with water? Yeah, those polar ones. Polar covalent bonds. Polar covalent bonds. Those are in compounds that play nice with water. The reason they play nice with water is because they have dipoles. So here's maybe a big picture idea to write down for yourself. Compounds with dipoles play nice with water. Or compounds with polar covalent bonds play nice with water. Because water has these dipoles. The only kind of compounds that like to be around things that have dipoles are things that have dipoles. So things that play nice with water are things with polar covalent bonds. Polar covalent bonds, they have dipoles. If I don't play nice with water, it's because I don't have dipoles. It's because I am very happy in my chemical bond. I'm sharing my electrons equally and I have no charges. Which kind of bond was it again that we said we share equally or there's no charge? Which kind of bond was that one? Yeah, that's the nonpolar ones. Nonpolar covalent bonds. Those are the ones that do not play nice with water. Those are the ones that when we have a compound with this kind of bond, we're going to see that grouping together. We had that picture at the end of our time yesterday that said, I don't need that kind of negativity in my life, right? That's what these non-polar covalent bonds say. They say, you've got a charge, you've got a dipole. I don't need your negativity, or I don't need your positivity. I have no charge. I am happy where I am. So non-polar covalent bonds are in compounds that don't play nice with water. Last question we got over here is about electronegativities. So that was one of the other things we talked about yesterday. Remember when we talked about electronegativities, we said in easy person words, this is how badly an atom wants electrons, how badly it wants those electrons to be circling around it. So if I've got a very similar electronegativity with the atom that I'm bonded to, if we have similar electronegativities, we both want the electrons the same amount. And if we want the electrons the same amount, then we're probably going to share those electrons equally. When we talked about equal sharing, I keep asking the same question, right? When we talk about equal sharing, which kind of bond are we talking about here? Yeah, it's that nonpolar covalent bond. Nonpolar covalent bond. If we both want our electrons the same amount, we're going to share them equally. We're both pulling them toward ourselves equally. If we don't want them equally, if one of them wants it more than the other one, we're going to end up not sharing equally, forming a polar covalent bond. Later today, we're going to talk about if one of us just wants to give away the electrons and the other one wants them really badly, we'll actually form our last kind of chemical bond. Who knows from working on the lesson, what's that last kind of chemical bond that you're learning about? Yeah, so ionic bonds. Ionic bonds, that's the last thing we'll talk about today. I like to call ionic bonds stealing bonds. Stealing, can't spell. Stealing bonds. How we call covalent bonds sharing bonds. I'm going to call ionic bonds stealing bonds because in an ionic bond, somebody gets an electron, somebody gave away, or I had an electron stolen from them. So somebody's all the way positive, somebody's all the way negative. That's how we have an ionic bond. Hey, so one of my groups told me this first statement here was a little tricky. This kind of bond involves sharing protons. My second group had an idea on this one. One of my other groups had an idea. What is the correct answer for this one right here? Which kind of bond forms when we share protons? Yeah, none of them, none of the above. Actually, no kinds of bonds form that way. So none. We are always sharing electrons. And that's what we emphasized in our class yesterday when we were talking about bonds. It's always all about electrons. Yeah, Jacqueline's like, I was so confused. Yep. I'll tell you guys when I write questions on your homework assignment or on exams, sometimes it looks painfully obvious that it's wrong. If it looks painfully obvious that it's wrong, it probably is. Like I promise that I write questions that are sometimes painfully obvious. So if you look at this and you're like, I don't know of any kind of bond that forms from sharing protons. Well, that's because it doesn't exist. So you were 100% right when you looked at this and you're like, this does not sit right. It's not about those protons. It's all about those electrons. That's the only thing I can share. Yeah, Karina's absolutely right. The protons are in the middle of an atom. We cannot share them. The protons got to stay where they are. They have to stay in the nucleus. So none of our bonds form because of sharing protons. We always form them because we're sharing electrons. So hopefully that was some good review for you guys of those types of bonds, the way that they work. We're going to build on these ideas a little bit during our time today. So I want to take you back to a statement that I think you had in the guided lesson that you have on your notes outline, and we mentioned this last time. We mentioned that we form the plasma membrane of a cell, or here's the terminology we mentioned yesterday about it, the phospholipid bilayer. We form this structure of the plasma membrane, phospholipid bilayer, because of the way that these molecules in it interact with water. So here's some things with water that help it to impact the way that the plasma membrane forms. When we talk about water, we'll do some true and false here. When I talk about water, water does or does not have dipoles. Well, here's a statement. Water has dipoles. True or false? Water has dipoles. What do we think? Yes, that's a true. So water has dipoles. When we talk about something that has dipoles, I guess this isn't a true and false, sorry. When we talk about something with dipoles, what kind of chemical bond does something have if it has dipoles? What was the type of chemical bond that makes dipoles? Yeah, it's polar covalent, right? So water has polar covalent bonds. Let's add another word to our list here. When we talk about water, we would say that water is polar. this was something that someone mentioned in the chat before we started our class today. There are two words we can use to describe compounds. We can either call them polar, like we see with water, or we can call them nonpolar. And so to going to our thought on the last slide where we're like, it seemed like it's a tricky question. Hey, watch, I promise this isn't a tricky question. If I tell you a compound is a nonpolar compound, which kind of chemical bond do you find inside it? I promise it's not a trick question. If I call this a nonpolar compound, yeah, it's going to have nonpolar covalent bonds. Water is a polar compound. We said that water has polar covalent bonds. So when I talk about a compound, if I tell you on a homework assignment or on the exam that something's polar, you know what type of chemical bonds it has inside of it. It has a polar covalent bond. It's a polar compound. And hey, if we've got polar covalent bonds, that means we have dipoles. Okay, so let's go back to our statement here. The plasma membrane, or the phospholipid bilayer, forms because of interactions with water. So there's something about water, the fact that water has dipoles, and that it has polar covalent bonds, and that it's polar. All of these characteristics are going to drive us to form this thing called the phospholipid bilayer. So let's look back at the structure of phospholipids. Remember, this is where we started yesterday during our class. We said that each phospholipid in the membrane, it has phosphate, we're going to call this the head group. Head group. Phosphate head. And then we have those lipid tails down there. They're labeled, but I'll label them again, the lipid tails. When I talk about this head group up here, the question was asked yesterday, do I need to know the chemistry of all this? No, we don't need to know all of this. What we do need to know, we've got a phosphate group in here. And notice that this phosphate group has this negative charge portion on it. This negatively charged portion up here is a portion that would be very happy to be next to something that's positive. Something like a positive dipole that I find in water, for example. This head group portion of my phospholipid with its phosphate, with that negative charge on it, this portion of my phospholipid plays nice with water. So this top part up here with its negative charge plays nice with water. Now I know it was a really long, complicated word, but can someone help me out in the chat? Do we happen to remember what the technical word is for play nice with water? Plays nice with water. What was the technical word for that? Yeah, so it's a long one. Hydrophilic. Hydrophilic. You can break it down though. The first part's easy, right? Hydro. That means how do I feel about water? This philic part technically means that I love water. Philadelphia is the city of love. So hydrophilic means I love water. This top part up here of my phospholipid, the head group, is considered hydrophilic, meaning it loves water. It wants to be by water. Hey, the reason that it loves water is because it is polar, meaning it does not share its electrons equally. That's why I've got this negative charge over here. This portion of my phospholipid is polar, which means that it plays nice with water. So in this lesson, you're going to see a lot of synonyms, words that mean the same thing. Two words that mean the same thing, hydrophilic and polar. These words are synonyms for each other. But these words means that we play nice with water, that we like to be around water. When we go down here to talk about the lipids are considered nonpolar. They're nonpolar. I'll do the whole thing in caps. Nonpolar. Nonpolar, remember, means that we share our electrons equally. Whenever we're talking about something that's lipids, or lipids, remember, is a fancy word for fats. Whenever we're talking about fats, like we see down here, they have nonpolar covalent bonds. They share their electrons equally. So when we talk about things that share electrons equally, that have nonpolar covalent bonds, don't play nice with water. They don't like water. Can you help me out in the chat? If I don't like water, what's that technical word now? Does not play nice with water. Yeah, that's the one that maybe is easier for us to figure out, right? Hydrophobic. Not only do I not play nice with water, I'm actually afraid of it, a phobia of water. Hydrophobic and nonpolar are synonyms. So here, let's add that over here. Hydrophobic plus nonpolar are synonyms, just like hydrophilic and polar were synonyms. So I've got this molecule right here. This is a phospholipid. Here, I'll add that title. This is what a phospholipid looks like. Two parts, the phosphate part and the lipid part. There's a word that I think you see at the very end of this lesson, and that word is called amphiphilic. Amphiphilic is kind of like an amphibian that can live in the water or it can live on the land. It can do both things. We would call a phospholipid amphiphilic because it has one part that's hydrophilic and one part that's hydrophobic. So this molecule, a phospholipid, has part of it that loves water, that's this part up here, and part of it that is afraid of water, that's this part down here. So when I talk about this molecule of a phospholipid, the thing that makes up the plasma membrane, the thing that makes up the plasma membrane is amphiphilic. Part of it loves water, part of it hates water. And that's how it's going to contribute for us to this phospholipid bilayer shape that we see. I have a question in the chat. It says, so the head has a positive and negative charge, which makes it polar. That's correct. Yes, the fact that we have these parts up here that have that negative charge, and for that matter, I know we're not focusing on this glycerol right here, but I'll just tell you this bond right here, where I've got a carbon with an oxygen, these are also polar covalent bonds. So I've got all kinds of polar stuff going on up here. So the fact that I've got this negative charge, the fact that I've got some other polar bonds in here, that's what makes this whole area polar. There's a whole lot of unequaling going on. Absolutely. Okay, so these groups of words, hydrophilic and polar, and hydrophobic and nonpolar, underline, highlight, star, or make sure we know that these words mean the same thing. To help us remember that these words mean the same thing, let's go to that image of the plasma membrane that you have in your lesson outline. Can somebody help me out in the chat? What page are we on? Do we have an idea of where we are in that packet? Okay, I'm here on page 6. Perfect. Okay, so we're on page 6. What I want us to do together is we're going to work on labeling different parts of the plasma membrane using those words that we just talked about. So the words that we're going to use here, we're going to use the words hydrophobic and hydrophilic. Actually, here, let's do this smart. Hydrophobic areas, we're going to start over. Here we go. I want my hydrophobic areas. We're going to color them orange, hydrophobic. We're going to color our hydrophilic areas red. We're going to color our areas that have water blue. We're going to color our polar areas, green, and our nonpolar areas, purple. Okay, this is probably going to descend into mass chaos, but I don't even care. We're going to give it a try. If you look above the screen, there's a pencil up there. When you click on that pencil, over to the right of it, there is a circle that says colors. Pick one of those colors, and I want you to draw on this picture for me. We want to fill in some of these areas with these different color words. So let's see if we can, as a class, do some coloring here. Okay, I'm seeing a lot of my colors. There we go. I was going to say, we're all hating on green. Awesome. Here, while we're at it, let me draw you your daily penguin. Man, I was so good at this at the end of the spring, and I still got to practice. We're failing here, this poor penguin. Hey, we'll talk about this later in the semester. Guy kind of looks like he might have hydrocephalus, water on the brain. This poor guy. Okay, there's my penguin art for us. All right, let's look at our beautiful artwork that we made together as a class. We should definitely frame this and keep it forever, right? Okay, so the first thing that we worked on this picture here is we wanted to identify where there was water. And we actually did this during class yesterday. We talked about this. I've got a bunch of water in the very middle of my cell. Remind me in the chat, the water that's found inside the cell, all the soupy stuff in here, what's this stuff in here called? Yeah, this is my cytosol. So cytosol has a whole bunch of water in it, lots of water here on the inside. Then on the outside, I also have water. So we found that blue water here on the outside as well. There were two types of fluid that's outside the cell. What if I am in tissues? What's the fluid outside the cell called if we're inside tissues in the body? Yeah, that's that interstitial fluid, right? The one that's hard to spell, interstitial fluid. And if I'm inside a blood vessel, this stuff out here, what's that stuff called? Yeah, that's the plasma. Perfect. Okay, so here on the inside, we've got some watery cytosol. Here on the outside, we've got either watery plasma or watery interstitial fluid, plus a penguin, right? For good measure, we've also got penguins in our bloodstream. We'll say, hey, this penguin, we can say, represents our immune system. He is going to beat COVID, this little penguin here, and let me tell you, we want him in our bloodstream. All right, here we go. So plasma on the outside, cytosol on the inside. That's our blue stuff. Notice that next to the blue, I have green on the inside, or I have red on the inside. We just said this on our previous slide, that red, hydrophilic, and green, polar, mean the same thing. So we're absolutely right that we colored this inner ring here red and green. It's Christmas in our cell here. So red and green going on, polar and hydrophilic, touching this watery cytosol. Remember, hydrophilic things like to interact with water. This part of my phospholipid is totally cool with being next to cytosol. Here on the outside with my plasma or my interstitial fluid, notice that again, I've got red and green. Red and green, polar and hydrophilic, those were the words for things that do play nice with water. So I've got these head groups that do like water. They're polar or they're hydrophilic. They're right next to the watery stuff on the outside and the watery stuff on the inside. Then we look here in the middle of our membrane. Notice that the middle of our membrane here is colored purple for its nonpolar. It's also colored orange for being hydrophobic. These are the parts of my phospholipids called the tails. Remember the lipid tails? this part of my plasma membrane inside here does not like water. Notice that we didn't put any water in this middle part right here because there is no water in this middle part here. I'm gonna make everything go away. Let's all be sad for art. Here it goes. Well, I mentioned for you, these little lipid tails inside here, they squeeze water out. These guys, to use the word that we had on the other side, these are nonpolar. Nonpolar, in other words, means hydrophobic. I can't stand water. So nonpolar and hydrophobic, that's this middle part right here. From your lesson, you saw a little picture then of a bouncer that's inside here. The middle part of the plasma membrane with these lipid tails, that's what's going to decide what can and cannot get through. Help me out in the chat. Do you have to match or be different from the chemistry of these tails to be able to get through? The stuff that can go in and out of the cell, does it match these tails, or is it the opposite of these tails? Yeah, so several of us are chiming in, albeit hesitantly. Yes, you have to match these tails to get through. So these tails are nonpolar, these tails are hydrophobic. If you are something that's nonpolar, if you match these tails, the tails say, oh yeah, come right on in or go right on out. I don't care, you can move. If you don't match these tails, so let's use our non-matching words, we'll label them here. The heads were polar, or the heads were hydrophilic. Okay, that's the same thing here on the outside. The heads are polar, the heads are hydrophilic. If you're something that matches these words, if you match this part, the outside of the membrane, if you're hydrophilic or if you're polar, these tails say, you don't got the clearance, you can't come in. So things that don't match the middle of the plasma membrane, they can't pass straight through the plasma membrane, at least not without help. See, in lesson number 4, you're going to learn about transport proteins. Transport proteins basically make a bridge for things that are polar or things that are hydrophilic to go from the outside to the inside. It makes a passageway for them, because the only things that can actually get straight across the membrane are things that are nonpolar or things that are hydrophobic, things that match this part of the membrane. So that's a big, important idea to make sure that we understand. The part of the plasma membrane that decides what can get in and out are these lipid tails that are here in the center of the membrane. My head groups that I find toward the inside of the cell membrane or on the outside of the cell membrane, these are polar things, and they're not going to they don't play any role in determining what can get through, but they do help me to interact with the water on the outside and the water on the inside. I've talked a whole lot. Let me pause for a moment. Help me out in the chat. What questions do we still have? Or send me an emoji. Show me how you feel. Questions and emojis. Let's open up the floor. Ariel's looking like she's doing good. Nice, dinosaur. I have not seen that one yet. That's great. Jesse's good. Here's my penguin that doesn't have hydrocephalus. Yeah, so Vanessa asked, you'll see the terminology of biochemical properties. Biochemical properties. And that terminology, yes, that means is it polar or nonpolar? That's what biochemical properties mean. Is it hydrophobic or hydrophilic, which are synonyms to each other, right? Yeah, when I use that terminology, biochemical properties, that's what it means. What's going on with its chemistry that dictates its biology, the way that it interacts with things. So Nicole, just know that, because you might, I think that's in one of the learning objectives, actually. It asks you to explain how the biochemical properties of these parts of the membrane lead to what can and can't get across. So just be familiar with the fact that that's what I'm talking about when I say biochemical properties. That is it polar or nonpolar, hydrophobic, hydrophilic? So the tail will decide what can get in the cell. That's correct. Yep, it's these tail parts that I find here in the middle that are forming the barrier to decide what can and cannot get through. The head groups don't play a role in deciding what can and cannot get through, but they're important because these head groups like to be in contact with water because they're polar. So they're actually there. You could think of them kind of as a protective layer for these tails, because these tails can't stand water. They just can't even with it. So let's put a head group on the outside and a head group on the inside of the membrane to allow these guys to not have to deal with water or things that make them uncomfortable because they're not nonpolar. Good questions. Any other questions? You're welcome. Okay, well, let me show you an application of this. Oh yeah, so here, this is actually probably where that question came from. Should have just gone one more. You've got this critical thinking question, right? So it says based on the chemical properties, or it might say based on the biochemical properties of the middle of the membrane. Let's summarize. There are two words I use for the kinds of things that can go straight across the membrane. What do I call the kinds of things that can go straight across the membrane? In other words, what are the words that I use to describe my tails? There were two words for these tails here. Yep, so the first word I use to describe things that can go straight across the membrane is they are non-polar. They share their electrons equally, no charges. And then the second word that I'm seeing pop up in the chat some too is they are hydrophobic. They're afraid of water. Because remember, these middle parts here, my tails, they're afraid of water too. So things that are non-polar or things that are hydrophobic can go straight through these tails. Absolutely. Nicole mentioned small things. That is correct as well. Some small things can squeeze through. For the most part, though, what we're the two main ones that we want to make sure we know are things that are nonpolar and things that are hydrophobic. Generally, they got to be small nonpolar or hydrophobic things, because if we're talking about a really, really big nonpolar thing, then that wouldn't squeeze through because that would disrupt the membrane. But small nonpolar things, small hydrophobic things, those can pass straight through the membrane because of those tails. So here's an application for you of why it's good for us to have nonpolar things that can go directly in and directly out of our plasma membranes. The example here we're looking at is the process of respiration. And we're not going to cover respiration in depth this semester because that's an A&P2 topic. But the process of respiration can happen as easily and quickly as it does because the two things that are moving during respiration are things that are non-polar. So when we talk about oxygen that our cells need, that helps them to breathe, just like we breathe, oxygen has two oxygen atoms in it. And so these two oxygen atoms have exactly the same electronegativity because they're literally exactly the same. They're sharing their electrons with each other equally. This is a non-polar compound, oxygen, right here. Carbon dioxide, the fact that I have two oxygen atoms on either side of my carbon, means that I end up balancing the way that I'm pulling those electrons. So carbon dioxide is another non-polar compound. Now, when we talk about the process of respiration. Respiration is an example of diffusion. Respiration is an example of diffusion happening. What are some of the things, when we talk about the process of diffusion, what are some of the things that you guys know about diffusion? What are some of the most important diffusion ideas? How would you describe it? Okay, love it. One of the most important things for us to know is that this is an example of passive transport. So passive transport means that there's no energy needed, For it to be able to do passive transport for no energy required, several of us have honed in the fact that we go from high concentration to low concentration. We go from high to low. And absolutely right, at the end of this process, we end up with isotonic solutions. Love it. Okay, respiration, breathing, helps us, if we know that it's diffusion, we know that all of these rules apply. All of this is true. So the process of getting oxygen into your cells or getting carbon dioxide out of your cells, no energy required. That's good, right? Because we don't want to have to fight to get our oxygen. We also know, though, when we're looking at how this process works, oxygen is always going to move from where there's a lot of it to where there's less of it. The reason that this statement right here is helpful for me is when we look at this picture here, notice that we start in the lungs. In the lungs, we breathe in and oxygen comes in. As we breathe in that oxygen attaches to the hemoglobin in our red blood cells. Now, based on the fact that respiration is the process of diffusion, so based on the fact that things go from where there's a lot of them to where there's a little of them, would we say that the red blood cells or the lungs have a higher oxygen content? Based on the direction oxygen moves, where do I have more oxygen? The lungs or the red blood cells? Yeah, so a couple of us have chimed in. We start with high oxygen concentration in the lungs because you breathed in. Lots of oxygen. That oxygen, there's a bunch of it in the lungs. There's not as much of it in the red blood cells. In diffusion, things go from high concentration to low concentration. So oxygen naturally says, we don't want to chill out in the lungs here. I want to cross the plasma membrane of a red blood cell and go inside there because there's not as much oxygen inside there. So the first part of respiration, we take that oxygen that's in the lungs, put it into the red blood cells. Second part is those red blood cells then travel throughout the body and they go toward organs. Organs like my favorite system here. We've got, let's see if we can identify. Help me out in the chat. I see 3 organs here. Can we identify what organs we see here? Let's draw some arrows. I got 1 organ right here. I got 1 over here. And I have 1 more down there. Okay, so several of us have identified the liver. That's my big brown one, liver, okay. And then, yep, some of us are identifying that this pink one over here is the stomach. My yellow one down here, Vanessa identified it. This yellow one down here is the pancreas. Pancreas, I guess someone mentioned the intestines. This is the beginning of the small intestine right here. So the pancreas is right up next to that small intestine. So we've got the big liver, the stomach, the pancreas. These are an example of some of the organs in the body. Red blood cells are going to travel past all of them. Yeah, we do have a little bit of the gallbladder too, peeking down there. Red blood cells go everywhere in the body. They're going to go next to all of the different organs in the body. But what always happens is that oxygen in the red blood cells leaves the red blood cells to go into the organs. If oxygen goes from the red blood cells into the organs, where do I have a higher concentration of oxygen. It goes from the red blood cells into the organs. Who's got more oxygen? Yeah, we're always going from high concentration to low concentration. So the red blood cells have a whole bunch of oxygen. They give that oxygen to the organs because the organs don't have very much because they used all of their oxygen to make energy. So here's how we could rank our oxygen levels. Highest in the lungs. From the lungs, we put all of that oxygen into the red blood cells. It was lower in the red blood cells. Then the red blood cells go out to the body, and the body is using their oxygen like crazy. There's more oxygen in the red blood cells than there are in the organs. So highest concentration of oxygen in the lungs, then the red blood cells, then the organs. When the organs are done using this oxygen that they got from the red blood cells, they use that oxygen to build ATP, to build their energy. Then they spit out carbon dioxide. So at the end of the process of building energy, we spit out carbon dioxide. Carbon dioxide is also nonpolar. So carbon dioxide goes from the organs into the red blood cells. We are still doing the process of diffusion, which means we're still going from high concentration to low concentration. So I've got a lot of carbon dioxide in these organs that I give to the red blood cells, meaning there's more in the organs than there are in the red blood cells. I transfer it. Interestingly, by the way, this carbon dioxide actually also attaches to hemoglobin, which is the same thing that oxygen attaches to. So it's attaching in the same place. So my organs have a lot of carbon dioxide. They give that carbon dioxide to red blood cells. Red blood cells go up by the lungs where we're breathing out, and we can actually expel that carbon dioxide because there's more in your bloodstream than there is in the environment. So we spit out that carbon dioxide to get it back out. Ariel's mentioning there might be a typo in the guided lesson. I will look into that, where it might say that the organs have more oxygen. So I will make sure to check on that. The organs don't have more oxygen, but help me out in the chat. They have more of something. What do the organs have more of than the red blood cells? Yeah, the carbon dioxide. So I will look into that and make sure that that gets switched. That's probably a typo. I apologize for that. More oxygen on my list to look into. So here's big picture idea. A couple of big picture idea takeaways. First thing, we're really glad that oxygen and carbon dioxide are nonpolar because that means they can go in and out of the membrane with no problem. The membrane lets them go straight through. The other thing with respiration is because these are nonpolar things that are moving with diffusion, they're always only going to be able to go from high concentration to low concentration. That's the only way that this process works for us. There are things that cannot move in and out of the cell through diffusion. You guys told me on the last slide that diffusion makes things isotonic, makes them match. But we know, especially, let me highlight the ones that we've focused on here. We know that there are several things that diffusion does not work on. It's those things we were memorizing for our cell. So the only things you have to know, the concentrations of here, are sodium, chloride, and potassium. We know that there's different concentrations inside cytosol, intracellular fluid, compared to outside. And the thing with all of these particular atoms that we're looking at down here, all of these are called ions. They're all called ions. What is a quick definition of what makes something ion? What is an ion? As opposed to just a regular atom. Any ideas?
It's an atom which gained an electron.
Absolutely. Yep. It's an atom that gained or lost an electron. So gained or lost electron. And that means that it has either, if we're talking about gaining an electron, a negative charge, or it has a positive charge. It can have either of them. When we talk about ions, we've gained or lost an electron, we have some kind of charge. Things with a charge do play nice with water. They like to be with water. If we like to be with water, that's another example of something that is hydrophilic. And hydrophilic things do not cross the membrane. They can't cross the membrane. They're usually dissolved in water, and water can't get across the membrane. So my things that are more concentrated inside or outside, a lot of them are ions. They're things that have a charge. Things with a charge cannot go across the membrane. They're stuck on the inside or the outside. Now, we'll talk about in lesson number 4 how we use proteins to get them across, but they can't do diffusion. They can't do what oxygen and carbon dioxide did. So let's talk about briefly how I make these ions. We make ions, like Mary mentioned for us, we make them by moving electrons. So here's a little dot that represents an electron. What happens is when we put together two atoms that have very different electronegativities, here's an important idea for us, very different electronegativities. When we put those things together, what happens is one of those atoms, this sodium, for example, says, I don't care that much about my electron. Meanwhile, chloride over here says, man, I really want that electron. Chloride steals the electron from sodium. Now chloride has an extra electron, an extra negative charge, so it looks negative. Sodium lost one of its negative charges. That makes it look positive. When we talk about the way that sodium and chloride interact with each other, it's that kind of bond we mentioned at the beginning. We form an ionic bond. An ionic bond is what happens, literally the basis of these bonds is opposites attract. Opposites attract. So we've got something with a positive charge. We've got something with a negative charge. The only thing that holds these two things together is the fact that you're positive and I'm negative. Since you're positive and I'm negative, we're attracted to each other. We're close to each other. So here is an important idea for you to write down or make sure we know about ionic bonds. In ionic bonds, we do not share electrons. We do not share electrons. We steal them. We steal them. Like I said, I like to call ionic bonds stealing bonds. Somebody has the electron. Somebody lost the electron. There's none of the going back and forth where they share the electrons. That's what makes this kind of bond weaker than our covalent bonds. Because in this kind of bond, we're not sharing electrons. We're transferring them from one atom to another atom. So ionic bonds are not sharing bonds. They're stealing bonds. And my electrons actually move. So Jacqueline's mentioning some of the terminology that you'll see in the lesson. We have the terminology of a cation, that's the thing with the positive charge, and an anion, the thing with the negative charge. So cation with a positive charge, anion with a negative charge, because electrons moved. The electrons moved and generated these two types of ions, the cation and the anion. Do not, let me say this now, do not memorize these numbers. I don't think you even actually have these numbers. But the reason I included the numbers on here was just to show you something. When we talk about a nonpolar covalent bond, those are my equal sharing bonds. The electronegativity differences, that's what this number shows me. My differences in electronegativity is pretty small when we're talking about a nonpolar covalent bond. For us to share our electrons equally, our electronegativities are pretty close to each other. If we kind of have a difference in our electronegativities, if we're somewhat different from each other, but we're still not like completely different, we'll form this polar covalent bond. And in a polar covalent bond, remember, somebody gets those electrons more often than the other person in the bond. So polar covalent, we're not sharing equally. Our electronegativities aren't completely matched. They're kind of different. Non-polar covalent bond, those electronegativities are very similar to each other. Notice in an ionic bond, here's what I want to highlight with an ionic bond. See how I've got one electron cloud and another electron cloud. In an ionic bond, we don't share electrons. I can tell because I have two different electron clouds here. One of them for my anion, that's really big. One of them for my cation, that's small, because this one gave away an electron that's now zipping around just this nucleus over here. As opposed to with my polar covalent bond, where my electron maybe does 2 or 3 loops over here, and then does a quick loop back over on this side, and then does another couple of loops over here, and then does a quick loop here. Electrons spend more time around one nucleus compared to the other in a polar covalent bond, but it still ultimately goes back and forth. When we talk about these ionic bonds, you're either circling this nucleus or you're circling this nucleus. You're never going back and forth between them. That's the biggest difference between a covalent bond, a sharing bond, and an ionic bond, a stealing bond. So make sure we know that we have two different electron clouds here because we're not sharing electrons. And the reason we're not sharing electrons is because our negativity, our electronegativities are very different. If they're very different, we make an ionic bond. If they're kind of different, we make a polar covalent bond. If they're about the same, we make a non-polar covalent bond. We share our electrons equally. Help me out in the chat. Questions or thumbs up? How do we feel about our 3 types of bonds? Yeah, so Jacqueline's asking about water. That's where we're going next. Nicole asked on the test, will we have to know the electronegativity? We don't have to know the numbers. We do have to know that in an ionic bond, electronegativity is really different. In a non-polar covalent bond, it's basically the same, and it's kind of in the middle on polar covalent. So don't memorize numbers. I'm never going to give you numbers. That's your chemistry teacher's job. I'm not going to do numbers. We just need to know generally very different for an ionic bond, not different, non-polar covalent, kind of different for polar covalent. And Nicole's asking if we need to know the colors on the table. I will give you a table in color if we're doing that on the exam. So no, you don't have to memorize those colors. All right, so we had a question about what this means with water. So let's talk about that. In my picture here, I'm looking at a spoonful of salt. If you pour that salt into water at home and you stir it up together, first thing that'll happen is you will magically transform your cup into a beaker, right? I need some more beakers at my house. I probably need to do this experiment. What will actually happen though, you probably won't get a beaker. Although if you do, let me know, because that's pretty awesome. What will actually happen is that salt will completely dissolve and you won't be able to see it anymore, or a measuring cup, there you go. You won't be able to see it in your solution anymore. So here's what's actually happening when you put salt into water. Salt is made out of sodium and chloride. So sodium is my cation. It gave away an electron, that makes it positive. Chlorine or chloride is my anion. It got an extra electron. So what ends up happening is the negative in chlorine is attracted to the positive in sodium. And the positive in this sodium is attracted to the negative in this chlorine. So you build these salt crystals where negatives in chlorine are attracted to positives in sodium. So the only thing, here's a reminder, here's what we need to remember. The only thing holding together ionic compounds is the idea that opposites attract. This is an underline highlight star idea. The only thing that holds together this sodium and this chloride is the fact that you're negative and I'm positive. It's just the fact that we have opposite charges. Because see, that gets really important when we're talking about putting this salt crystal in water. Remember, water has a partial negative charge and partial positive charges because it has dipoles. If I put an ionic compound into water where I've got partial negatives and partial positives, and I let those water molecules go around my salt, what ends up happening is this sodium that used to be happy with all the negatives in chloride, this sodium says, you know, oxygen's negative too, and that's pretty attractive. I'm interested in that. This chloride here that used to be happy with the sodium's positive charges, this chloride says, you know, hydrogen has a positive charge too. I'm attracted to that. So what happens when we put a salt crystal, so something with an ionic bond, in water, suddenly I have new positives and new negatives that I'm exposing these ions to. And when they see those new positives and those new negatives, I mean, they're not really in a committed relationship. They're just in an opposites attract situation. So opposites attract, well, I used to attract to you. Now I attract to the water that's around me. We talked yesterday when we talk about a bond dissolving in water. When we talk about a bond dissolving in water, that means that the atoms are no longer attached, They completely fall apart from each other. When I look at my example here where we started with sodium and chloride attached to each other, at the end of this scenario, sodium and chloride are not attached to each other anymore. Ionic bonds do dissolve in water. So that's a big idea for us. Ionic bonds dissolve in water. And remember, that terminology dissolve means that they completely fall apart. Sodium is no longer attached to chloride because sodium is now surrounded by water and chloride is now surrounded by water. An ionic compound dissolves in water. Now here's a reminder for us. Polar compounds do not dissolve in water. We talked about this yesterday, and I want to make sure that we understand this idea here. Compare this picture, what I want you to do when you're studying. Compare this picture of what we see happening with salt and water. Compare this to what we showed when we were looking at polar compounds. Polar compounds will surround themselves by water, but they're not going to fall apart. Since they don't fall apart, we would say that they don't dissolve. So please make sure to compare this picture to that picture that we looked at yesterday about what polar compounds do in water. Really fast though, I want to look with you at this. We're looking at what happens if you were to mix oil with water. So if I poured oil into water, stirred it up with a spoon, and then let it sit, I would end up with that oil sitting on top of my water. Oil is an example of a nonpolar solution. Oil is nonpolar. Help me out in the chat. What was that other word that meant the same thing as nonpolar? If something is nonpolar, how does it feel about water? Nonpolar equals, exactly, hydrophobic. Hydrophobic, as in I push water away from me. You let it sit for a long time, there's going to be no water here in the middle of the oil. All of the water is going to be down below, and all of the oil is going to be squeezed together. That's because oil is nonpolar, or it has nonpolar covalent bonds in it. It wants nothing to do with water. It's afraid of water. So by by squeezing together, it can push all of the water out. So here's a little cartoon that I want to end our time with today. So water comes up to oil, says, "Hey, oil, want to hang out?" And oil says, "I can't mix with you guys." Water says, "Hydrophobe." It's pretty accurate, right? Because oil is hydrophobic. Hydrophobic. So big ideas as we're working on our stuff with the membrane. Some parts of the membrane are hydrophobic. They can't stand water. Some parts of the membrane can stand water. They're not hydrophobic. The parts that are hydrophobic won't have anything to do with water. They get to decide what comes in and out. But it's important to note that if you're a compound that does play nice with water, if you like to be around water, the middle of the membrane is going to say, "I can't mix with you. You're not passing through. You're stuck on the outside or on the inside." So that's our ions that we talked about. That's things that are polar, like we'll talk about next week, things like glucose. If you don't play nice with oil, you're not passing through the membrane. So Jacqueline asked, so what would you need for oil to separate? There's some things that are called emulsifiers. I'll type that word over here. You don't have to know this word, but just toss it out there. Emulsifiers. You can use that to make oil separate. Even that's kind of the job of soap, for example. Yeah, so Nicole is mentioning Dawn dish soap and soaps in general. Tend to be emulsifiers, where they'll help to take something that's hydrophobic and allow it to mix with water so that you can get it off of your dishes. So yeah, emulsifier is a good example of that, is our soaps. Absolutely. Yep. Good application of our stuff, right? Dawn dish soaps reminds me of my sink full of dishes. Just being real, right? We are about out of time. Before I turn off my mic, are there any last minute questions today? Nicole asked, do we use the terms cation and anion when describing dipoles? Great question. Let me ask the class, what do we think? Cation and anion, is that the same as dipoles? Any ideas? Yeah, it is not the same. That's an excellent question. When we talk about dipoles, remember that dipoles are a partial charge. When we talk about a cation or an anion, that is a full charge, meaning it is always positive or it's always negative. Dipoles are only sometimes positive or negative, so they are different from each other. So good question. Cation and anion only used for things that are always charged. Dipoles used for things that are sometimes charged. Any other last minute questions? Yeah, so the things that I use to make an ionic bond are things that are fully charged. We use the cation and the anion to make an ionic bond. We use the dipoles to make those hydrogen bonds. So that's a good comparison to make. Dipoles are used to make hydrogen bonds. Cations and anions are used to make ionic bonds. Good comparison. Yeah, so the homework for this week is due on Sunday. That's correct, Sunday evening. Go for it, Miriam has a question.
Regarding the homework for this week, I know the unit one we're gonna be done with by the 20th. For the group wiki assignment, for unit one, was that the one that we've done at the beginning of the semester where we have introduced ourselves and stuff, or we still have to do something else plus the homework on Blackboard and a visible body?
Yes, so there's no visible body assignment this week or next week because they didn't have content that matched up well with what we were doing in class. So the next visible body assignment is in a couple of weeks. The unit number 1 or the exam number 1 wiki, that is different from what you've already done. So when you're in your group's wiki page, check over on the far right side of the wiki. There is a new set of questions posted for you. They are content questions. So that is different from the wiki you've already done. You can start working on that now. It doesn't have to be completed until the 20th, the evening before the exam opens, because some of the questions on there are going to come from lesson number 4. So just be aware of that, that you can start working on it now, but you probably can't answer all of the questions yet. And then Nicole asked if someone dropped out of the group or out of the class, do we do more than 3 questions? You're only required to do 3 questions. If you do more than 3 questions, that is worth a little bit of extra credit because it is extra work. So you only have to do 3. I would recommend making sure all of them get answered by the due date, just because it will help you study for that exam. But you only are required to do 3. And yeah, one person can do 3 questions. You could go in and do your 3 questions now if you wanted to and be done with it. But just do check back closer to the due date to make sure all those questions get done. Hannah has a question.
So you just said that the exam will open on the 20th.
The exam opens on the 21st, so it opens on the Monday. Yep.
Gotcha.
The assignments
Okay.
are due on the 20th. Yep. So the assignments are due on the 20th. The exam opens on the 21st, and it will be open through the evening of the 22nd. So you have a little bit of a window to take it in.
Gotcha. Okay, that makes sense. Thank you.
Mm-hmm. And yeah, Ariel asked if we will have a review for the exam. Yes, we will next week when we get a little bit closer to it. I am bumping up into sharing my room. So last question, the chemical bond that must be present for oil to separate. So oil has nonpolar covalent bonds in it. It's never that we're going to separate oil in the same way that we do in an ionic bond. So oil never dissolves. We can just make it go from being in a clump like we see right here to mixing it up a little bit better. That's what an emulsifier does, but oil will never actually dissolve in water. That's only something, like you mentioned, that ionic bonds can do. All right, unfortunately, I have to call it a day because, like I said, I'm sharing my office today with my husband. So I'm going to stick around in the chat for a little bit longer if you want to chat questions with me. But I'm going to turn off that mic and transition out of the live session. But have a great weekend. Feel free to email with any questions that you have, and I will get a schedule posted for office hours next week soon. So we'll see you guys again soon.